All right, well, as I was saying, we've heard a good number of talks today, and it's gone with, of course, the immediate unfortunate exceptions. It's gone almost exactly to plan. The end of our plan is that someone, I'm sorry to say it was me, was assigned to make concluding remarks and summarize talks and things like this. My head is swimming with all of the ideas that we've been treated to, and I think it's really presumptuous of me to try to encapsulate the sort of key ideas of so many good speakers. What I do plan to do is make just maybe a few general points, though I would say that one of Tamsin's last slides, her way forward, said much of what I was going to say, but I'll put it in different ways. And then just to give you this to look forward to, what we may do is quickly swing some of our chairs up here and ask all of our speakers to come to the front and make it easier for everyone to ask follow-up questions of our different speakers. So what in the way of observations or closing thoughts can I bring to this? I was struck at the beginning of this morning by Jim's plants and their interesting ontogenies, and how much we learned from looking at time series of what was happening in their environment and in their responses. And I have to say this matches some experiences of my own where I look at long time series of records within individuals and in the fossil record between individuals, sometimes between individuals thousands of years apart. And I've always been impressed at how much additional information there was in these longitudinal approaches, longitudinal in a couple different senses of the word and scales. It's more than just looking at a collection of values that show certain scatters, even if you're sensitive to variability, certain variability as well. The world often gives us patterns of change through time, and those patterns of change are critical. They're really part of the whole picture that we have to understand. Jumping from there, I'll just sort of move randomly to one of the last talks, Sean's modeling. I thought was a really impressive use of sophisticated techniques to take these complex patterns and integrate them over time, over space, and use them to generate insights that we really couldn't have had in the same way if we'd just sort of tried to stare at the puzzling patterns on maps without some more hypothesis-driven, more strategic plan for how we were going to ask questions of these data and get answers. There's been a lot of talks today that gave us information on new systems, be it fire or aspects of digestion. Again, I don't think I'll take on the role of trying to summarize or find the most interesting points of all this. I'll hand that off to you. I think what we as students, as practitioners, as teachers of these sorts of topics need to remember is just the degree to which the natural world is complex. It is multiscalar, and we do, as Tamsin said, have to embrace it in all its complexity in order to really hope to not just understand, but correctly represent sort of the structure of our thinking and our advice to our students about new ways forward in addressing such topics and such general systems, such general thoughts. I think I'm going to leave it there. Let's a few of us can bring up some chairs, and I invite the speakers to come up front and let us pepper you a little bit more, particularly since we are bound to stay put for the time being. That looks like it.
Thanks,
We should, yeah.
Check 1, 2.
If people would
check,
perhaps
check.
address their questions to either one of the speakers or just to the group as a whole, feel whatever works best for you. Let's do that. In other words, I don't want to manage it too much or get in the way. But I think even though we are constrained to be here, this is a good opportunity that we should take advantage of. George.
But maybe it would be nice if we just asked everyone who wanted to contribute their views on what today brought to you that you didn't know last night.
Good suggestion.
Hello? I'll make a suggestion. The field is alive and well. I am so impressed by the young people that made presentations today. The level of sophistication, the breadth of the questions is truly amazing, and it has been a real pleasure to listen to the talks.
Oh, okay. Yeah, I think that the breadth of applications and the different new methods that are continually that people have continue to come up with to address some of these outstanding questions in respective fields always blows my mind away. I mean, isotopic systems are just, basically we talked about carbon, nitrogen, and strontium today, and that just scratches the surface even of isotope systems, and it just makes me excited to see where everything is going in the future. And for me, getting a more of an ecology perspective is always really nice, because I feel like I sometimes get a little sequestered, so it's always interesting to hear multidisciplinary perspectives on some of these issues. So. Oh, that one.
I mean, I guess being in this career is really satisfying in the sense that it is on. Hello. Good. All right. Being in academia is actually quite satisfying. I don't think any day goes by without me waking up and learning something completely new the next morning. And today it's just the same, basically the same principle, just applying over and over again, from being able to use isotopes and connect them to these spatial tools that allow us to identify places that are important to take care of, from learning about new ways of measuring nitrogen fixation. So those are things that I just woke up today and I learned about it. So it's really fascinating.
I agree with Alison. The breadth of applications are impressive, and I like that. Many of us have similar questions in completely different systems, and we have the same concerns sometimes. And it was nice to also hear Tansi's talk and to realize that, yeah, I'm not crazy. Like, my concerns are valid, real. I don't know, like, I enjoyed each one of the talks. I learned so much today. Yeah, thanks for having me.
Yeah, something I was just discussing with somebody at lunchtime is that the field of biogeochemistry in particular, like, you look at people doing molecular genetics and they're sequencing all these things and making all these amazing trees, and I feel like our methods by and large are so far behind that in terms of a lot of things we can do. But where the methods really are developing, you know, I think there are a couple of kind of niche spaces, and isotopic analysis is really one of them, particularly, you know, the kind of stuff that Pete was talking about, sort of position-specific gas isotopes, attributing that to different processes. It's really one of the areas I think that's pushing biogeochemistry forward the most. And also, yeah, I was just overwhelmed by all of the fun, cool things, and the posters in particular, mammoths, and it's just exciting. Like, it's a fun time to be doing this as a scientist.
So I share all of those sentiments. I originally got interested in isotopes working in Jim's lab for this particular reason, because of, well, when I was a young person in science, I didn't want to get pigeonholed doing one thing. I wanted to be able to do lots of different things. And this is a great example of how, by becoming trained and versed in a set of tools, knowing you know, that you can apply it in multiple situations across different scales. So it was really great for me to see how, to spend all day talking about how isotope variation can answer questions from, you know, deep time to daily, you know, small spatial scales to large spatial scales. So, yeah, that's what I think.
Yeah, I think we're now, because I'm the seventh in line, but it's the diversity for me was, I think, the most impressive, to really see how different, the same methods can kind of be applied to so many different things. For me, in many cases, it's really an eye-opener, actually, especially yours, on the spatial stuff. And actually, I realized I have other data, which I need to talk to you about, for instance. Apparently, I can do much more, so that's great. So that's what I learned from today.
I think one of the things that I really like about doing, using isotopic analysis, is that there's a community who use a technique, that we're united by our technique, but our research interests are so profoundly different. We're asking similar questions, but in totally different situations. And what that means is that you talk about it with people who have a completely different outlook to yourself, and it makes you reflect on your own questions and brings fresh insight. And I think that is really great. So no doubt all of us go to our own, you know, plant physiology conference or soil biogeochemistry conference, but there's also that kind of that perpendicular direction, which is extremely stimulating. So that's fantastic. Yeah.
I would just remind people of one important thing to remember, and that is the most important topic you will ever talk about in a day is the isotopic.
Right. Okay. Good.
Can I just share a brief update with you? So we've now heard that they can no longer confirm that there is an emergency issue, but that we're supposed to stay away from Mason Hall. So I'm glad that our worst fears aren't realized, and it looks like it was a false alarm. Okay, so carry on, please.
Another point?
Yes. Wait, sorry.
I've actually got this.
Oh,
If you have anything to say, go ahead, go for it.
oh, no.
Okay. I may have misunderstood George's suggestion, or in fact, I may not have, and here's another possibility. We've heard from the speakers. Are there young people from our community here that would like to say something about what they learned today, or sort of new perspectives that they've gained from coming to this meeting? If this doesn't raise a hand for you, or two or three, we'll move on. But if it does, that would be great.
Well, we can just ask some of our younger.
George is more forward.
Yeah,
about everybody without the word doctor in front of them has to say one thing?
Mess this up. Here you go. Come on, come on.
Well, while these guys are thinking about it, one of the things that struck me was that back in the early days of isotopes, if you had one isotope value, you could publish it because isotopes don't lie. And so if it showed this or if it showed that was great. And all of these talks, I'm just blown away by how many values, thousands of values of isotopes that really nail down both the main point and the general trend, but also the variation around it. And that's something that's just really, really cool to see for me.
I have a general question following what George was saying. How do you guys think you can, or can you maybe incorporate some of the older data or some of the data sets that have fewer data points and sort of acknowledging their shortcomings? How can you sort of move forward with them? Do we need to redo all of those analyses, or has this made you guys rethink of everything that you've done in the past as maybe insufficient, or sort of how do we move forward on that level? I don't know.
I was just going to say that, so the work I showed this morning is actually an extension of a study that my advisor published in 2001 that measured carbon isotopes and alkanes from the same section. And we've added to that data set, extended it, and built on the trend, and it becomes even more robust with more data. And I think that I think old data sets that, you know, we know more about them now, or we see that we didn't have the resolution to ask questions before, it's somewhere to start from and then continue to build on and ask more specific questions and add more information. So I don't think it's a detriment. I think it's something that we can use as a stepping stone. And I think a way forward, like one of my projects that I didn't talk about today is actually a meta-analysis of some modern data of a bunch of different studies that if you take all of them together and do additional work, then maybe you can parse out some questions that you might not have been able to, where you only have a certain amount of money to make a certain amount of measurements, and the more we study them, the more we get. And as a community, I think it's a really useful way to move forward. So yeah, I don't think it's a bad thing. I think it's just, you know, science is always progressing. We're always doing better.
If I can follow up from that, I think Jim said something really interesting and profoundly important when he said when he's at the public trough, he feels obliged to make the data available, which is absolutely right, but that you want to make all your data available. And I think we only get where we are by standing on the shoulders of giants and giant data sets, other people's data sets. And it's quite interesting that other communities that generate data in other ways now have very large data sets. So there is GenBank, there is Proteome, which is moving forward. And it's always surprised me that we don't have any very good or comprehensive isotope repositories. There's the IAEA one for oxygen and hydrogen in water. I like to irritate geneticists by saying, well, of course you can have a database because genetics are really simple and isotopes are really complicated, because we have to factor in things like different tissues and whether or not it's got lipid in it and, you know, what it's made of. But it is increasing over the last 5 years, a realization across whole different sections of the community that we need to be finding a better way to share our data. So there are various small-scale archaeological databases, there are radiocarbon databases that include isotopic data, there are spatial databases, and now there's a new one which is funded by NSF. I don't know if anybody's heard about Isobank, so that's with Gabe Bowen and Seth Newsome and Brian Hayden and various others, so that's starting out. But I think we really need to be thinking about how we pool our data in a way that lets you get access to other people's orphan data sets, because it's always the file drawer problem. You know, we all have some random samples that we ran and we don't know where to put them and we don't know how to publish them, and so we don't deal with them. But, you know, what if there were actually 27,000 types of butterflies that had been run in loads of different studies and nobody had ever got around to publishing because they were all doing them in 3? So we need to find some way to share that, and I think that is, to me, I think that will be the big change. That in 20 years' time, we will be better sharing our data, or at least I hope so, when I retire.
Just make one comment, 2 comments actually relevant to your question. And that is, those of us that don't know history are doomed to repeat it. Yeah. And the second is, there was actually life before Google. As an editor, I realized that we all want to cite the most recent papers, but there are classic papers, and they may only have 2, 3, 5, 10, 15 data points. They worked hard to get them. Those facts are still facts.
So while
Ian.
we are talking about data, I think one thing that struck me today was, in addition to all the exciting advances that we've made in isotope geochemistry in the past several decades, there's still so much to do with just basic environmental data. There's so much to collect. And, you know, on a different continent, how do animals do in the same forest, and going back to the same place for almost 40 years and see how things are doing. So that sort of data, and I can see all the practical challenges with, you know, collecting that kind of data. And I'm not suggesting that we should all go out and sample everything possibly every time, but it's not really a question, but just some thoughts that several talks gave me. It's just all of the basic data that we still need to try to acquire. And if I have to ask a question, I'm going to ask a question that my advisor often asks me. And it could be for any of the speakers. So if you could have all the data in the world, anytime, any taxa, or any environment, what do you think it would be? What's the data set that you want the most right now, or you think we should go get? It's a big question.
I don't have anything. I have a second stable isotope of phosphorus, but if we're limiting it to like the real world.
How about a strontium map of every river out there?
I guess I'll answer from my biased point of view, which is I think one of the pressing challenges right now is how we deal with climate change, right? So there's still much to be learned about processes that release greenhouse gases, and learning more about those and getting a full data set as to how we can really understand all those processes. I think it's key for our sustainability. So I would ask for those data.
And I feel like my brain is very dispersed, so I have like many projects going on right now, like, but I guess related to my enrichment project, like I will test if, like, as I expect, my predictions hold true. So I will like sample, I will like work with zoo animals and measure the enrichment between diet and tissues, like across like huge number of mammalian species, to see how correct were my regressions, for example.
I have a suggestion. We all learned a lot by the Zachos curve. We can go backwards. Where would we be today if we didn't have the Keeling curve? So I think the data sets that I would want, if I could have any data sets, are those things that don't get recorded in some material that I can analyze, that I wish 20 years from now, I wish I had today.
Yeah, great. Yeah, that's good. Other questions?
Well, maybe this is a good time to try to wrap things up. We do have word from the Department of Public Safety that there's no longer, we don't think there's a threat, although people are asked to stay away from Mason Hall. We're about, for those of you not familiar with the campus, that's about 3 blocks from here. So we are able to continue with our program. The next thing is for a reception, to which you're all invited, and I hope you can all talk with the speakers more. Before we leave for that, though, I'd like to thank a number of people. First of all, let's have a big round for all the presenters. And unfortunately, I think this is almost in absentia because I think that Molly and Katie and John and, oh, let's see, you are still here, Dale. Many of the folks who have made this work behind the scenes, let's give them a big round as well. And I'd like to thank all of you in the audience for coming, and particularly those who I realize have come from quite a distance. We have people here from Notre Dame, from Michigan State University, from Bowling Green State. What else have I missed? I saw Wayne State on a T-shirt, but I don't know if that's correct. What other places from afar do we have? Anything I haven't mentioned? Oh, there's Wayne State. Oh, okay. Well, welcome. Well, anyway, I think that's terrific, and especially welcome to all the students who have come, and hope that you've all learned a lot. And so we can wrap up the formal session now, but we've got, as before, the food and drink are in the side room, but once you get that, there are tables and chairs out throughout the atrium, and you're welcome to take your food and drink and go out and sit down and mingle with various people you'd like to talk to.
Catherine Badgley deserves a big round of applause for all of her work.